Which is the type of microscope that requires a specimen to be dead, thinly sliced and stained with heavy metals? The answer is D, transmission electron microscope, because it shoots a beam of electrons through a very thin specimen to see internal details, and this requires the specimen to be dead. We cannot choose the scanning electron microscope because the specimen is already dead but not sliced. Compound and optical microscopes are used to view live specimens. The photomicrograph shows which kind of cell? Of course, it's prokaryotic cell because it shows a cell lacking a nucleus and other membrane-bound organelles. Which of the following orientations of phospholipids best represents the phospholipid bilayer of the plasma membrane? Of course, the answer is A, because the plasma membrane is composed of two layers of phospholipids, and in each layer, the hydrophilic or water-loving phosphate heads face the watery exterior or interior of the cell, and the hydrophobic water-fearing lipid tails face each other in the middle of the membrane away from water. Which situation would increase the fluidity of a phospholipid bilayer? The answer is C, increasing the number of cholesterol molecules. Because cholesterol is unsaturated fatty acid, having kinks in their tails due to double bonds, and these kinks prevent the phospholipids from packing tightly together, which increases the membrane fluidity. Which is the key property of the plasma membrane? The answer is C, selective permeability, because it allows some substances to cross easily, like small nonpolar molecules, while blocking or controlling the passage of others, like ions and large polar molecules. Which is not a factor that affects the rate of diffusion? The answer is A, because all factors are affecting the rate of diffusion except conductivity. Which type of transport requires energy input from the cell? Of course, the answer is A, active transport, because it moves substances against their concentration gradient, and this requires energy or ATP. Which type of solution could cause an animal cell to burst? The answer is A, hypotonic solution, because a hypotonic solution has a lower solute concentration than the cell's cytoplasm, and water will move into the cell by osmosis, and an animal cell, which like a rigid cell wall, will swell and can burst, but the plant cell becomes turgid due to the presence of the cell wall. By which method of transport can large substances enter a cell? course, the answer is endocytosis, because endocytosis is the process where the cell membrane folds inward to engulf large substances. And you know that facilitated diffusion and osmosis are for small molecules and ions, and exocytosis is the process of expelling large substances out of the cell. Which structure is found in both plant and animal cells? The answer is ribosome. And ribosomes are non-membrane bound organelles responsible for protein synthesis and are found in all types of cells, both prokaryotic and eukaryotic. Which structure synthesizes proteins that will be used by the cell? Of course, the answer is C, ribosome also. Which is the site of protein synthesis? Of course, the answer is endoplasmic reticulum. The rough endoplasmic reticulum is the site of protein synthesis for proteins to be exported from the cell or embedded in membranes. And this rough endoplasmic reticulum, it's rough because it's studded with ribosomes, which are the actual molecular machine that build the protein.